In this video, we will talk about the different blotting techniques that are used to detect your macromolecule of interest from a mixture of that particular macromolecule, for example, DNA, RNA, or protein. So if you want to detect your target DNA from a mixture of DNA, you will use southern blotting. If you want to detect your target RNA from a mixture of RNA, then you will use northern blotting. And if you want to detect your protein of interest from a mixture of protein, then you will use western blotting. So all of these blotting techniques are used to detect your macromolecule of interest from a mixture of that particular type of macromolecule. The basic steps involved in all of these blotting techniques are same. So first of all, you will need to extract the macromolecules from the cell. For the purpose of DNA, you will need to extract a genomic DNA from the nucleus of the cell. And if you want to detect RNA or protein, you will have to isolate RNA and protein from the cytosol of the cell. So extraction is not the topic of discussion here. So we will move onwards towards the blotting technique. So first of all, after extraction of the macromolecules, you will have to load your sample in the wells in the gel and perform gel electrophoresis. The wells are towards the negative electrode, whereas the macromolecule will move towards the positive electrode. We will discuss the procedure in detail in next slide. So after gel electrophoresis, you will get bands of the macromolecules on the gel that you can visualize under UV light. After getting bands of macromolecules on the gel, you will need to transfer these bands to the paper because gel is fragile and it can easily break. So you have to create an exact replica of the bands that you get on the gel onto a paper. So that's why this technique is named as blotting technique, in which you have to create an imprint or exact replica of the bands you get on the gel onto the paper. So after transferring the bands from the gel to the paper, you need to apply a probe that can bind to your target DNA, RNA, or protein. So in case of DNA and RNA, you will prepare a probe that is complementary DNA, that is complementary to your target DNA and RNA, and the probes are mainly radiolabeled or chemiluminescence so that you can easily detect your target macromolecule onto that filter paper. In case of proteins, you will have to create a probe that is mainly antibody that can bind to the protein. And then after that, you will get an autoradiograph of the paper where you can visualize the location or position of your targeted molecule. So this was all about the blotting technique, general procedure that is followed in all of the blotting techniques. We are going to discuss each one in detail because there are few modifications that are used in southern blotting, northern blotting, and western blotting. So next, moving towards southern blotting. Southern blotting is named after Edwin Southern, who developed this method, and in this method is basically used to detect your target DNA from a DNA sample. So in this figure, you can see that the first step involved in southern blotting is the extraction of genomic DNA. As all of you know that genomic DNA is very lengthy. So you have to cut it into smaller fragments for the purpose of gel electrophoresis. For cutting the DNA into smaller fragments, you will need to apply the restriction endonucleases. Restriction endonucleases cut the genomic DNA at specific sequences into smaller fragments that can be used to perform the process of agarose gel electrophoresis. So in case of southern blotting, you will use agarose gel for the purpose of electrophoresis. So in this figure, you can see that you will load your samples towards the negative electrode. Okay, then you will apply an electric current. Because the backbone of the DNA is negatively charged, the macromolecule will tend to move towards the positive electrode. Okay, so the DNA fragments that have a low molecular weight will move farther apart in the gel, whereas the macromolecular DNA fragments that have a high molecular weight will not move farther apart. They will get stuck into the gel somewhere uh, in closer to the negative electrode. So the DNA fragments having low molecular weight will move farther apart in the gel. So next is southern blotting. Blotting means the transfer of the DNA bands you get on the gel onto the filter paper. So the filter paper we use in case of southern blotting is nitrocellulose or nylon fiber. Nitrocellulose filter paper is used to transfer the DNA bands from the gel onto the filter paper. Okay, so how do you transfer the DNA bands from gel onto the filter paper? So there is a specific arrangement that is used. So first of all, blotting buffer. You will place the blotting buffer in a beaker. The blotting buffer in the beaker upon then you will place a sponge. A sponge will be placed in the blotting buffer. On the top of the sponge, you will place the gel. You will place the gel, and on top of the gel, you will place the nitrocellulose filter paper or nitrocellulose membrane. On the top of nitrocellulose membrane, you will place paper towel, and then you will place a weight that might be a 0.5 grams weight. So this arrangement of the blotting buffer, the sponge, the gel, the nitrocellulose membrane, paper towel, and the weight, it will create a capillary pressure, due to which the blotting buffer will move from the sponge to the gel, and then to the nitrocellulose membrane, and then to paper towel. So how is the DNA transferred from the gel to the nitrocellulose filter paper? The blotting buffer, while moving from the gel to the nitrocellulose filter paper, will take the DNA bands along with it, and it will create an exact replica of the DNA bands that are present on the gel onto the nitrocellulose membrane. So in this way, an exact replica of DNA bands on the gel is made onto the nitrocellulose membrane. After that, after getting an exact replica, you will separate out the nitrocellulose membrane from that. Then, as all of you know, that DNA is double stranded, and our probe is complementary DNA that we have prepared, and it will bind to our target DNA. So if the DNA is already double stranded, then the complementary DNA cannot bind with our target DNA. So for this purpose, you will have to apply denaturation buffer. Denaturation buffer is applied onto the nitrocellulose membrane so that the DNA becomes single stranded, so that our complementary DNA probe can easily bind to the single stranded DNA. And the probe is basically P32, a radio labeled probe is used in this type of blotting. So the probe will then bind to our targeted DNA that you can see in this figure. You can see in this figure here, the darkest band that you can see here is our target DNA with which the probe has been uh, hybridized. So probe hybridization is also an important step with the help of which we can easily visualize our targeted DNA onto the filter paper. Next, after applying probe, we will expose the nitrocellulose membrane to X-ray film with which it will produce a glow because it is radio labeled. So we will easily visualize our targeted DNA on that nitrocellulose membrane. So this was all about the southern blotting. Next is northern blotting. Northern blotting is very similar to southern blotting, but there are few modifications that are used in this procedure. So this is named after its similarity to the first blotting technique, that is southern blot. So this method is used for the detection of RNA in a sample. So first of all, you need to extract RNA from the cytosol of the cell. After extraction of RNA, you will perform electrophoresis. So the thing is that RNA is present in the form of complex secondary structures in the cell. It is not in the form of simple linear structures. So you will have to perform the gel electrophoresis, that is a denaturing gel electrophoresis. So in this case, you are using agarose gel electrophoresis, but you will add extra formaldehyde in the agarose gel so that the complex secondary structures of the RNA are broken down into smaller fragments of RNA that are linear RNA strands. So that, and why are we using this method? So that our the complementary DNA that we have prepared as a radiolabeled probe can easily bind to the linear stranded RNA. So after performing gel electrophoresis, agarose gel electrophoresis, we will get RNA bands onto the gel. So these bands are then transferred to the nitrocellulose membrane. After transferring, the process of transferring the RNA bands from the gel onto the nitrocellulose membrane is similar to that used in southern blotting. So after transferring, the RNA is fixed onto the membrane by using UV or heat. So after that, we will apply the labeled complementary DNA probe onto the nitrocellulose membrane that will bind to our target RNA. So in case of southern blotting, there was a DNA probe hybridization, while in case of northern blotting, there is RNA DNA probe hybridization, because DNA can easily and specifically bind to our target RNA. So we are using complementary DNA probe, so it will bind to our target RNA, and then we will easily visualize our labeled RNA on X-ray film. Next, moving towards Western blotting. The process of Western blotting is a bit different from that of southern and northern blotting. In case of Western blotting, so talking about the naming, Western blotting is named traditionally or inadvertently due to the southern blot technique that was named after the Edwin Southern, who discovered or invented this technique. So the naming of all these technique has nothing to do with the directions. So Western blotting is a method that is used for the detection of our target protein from a mixture of proteins. So in case of Western blotting, first of all, that you need to extract proteins from the cell. After extraction of protein, you need to load your sample into the gel. So in case of Western blotting, you will use STS page. So STS page is basically sodium dodecyl sulfate polyacrylamide gel electrophoresis. So after performing the gel electrophoresis, you will have to transfer the protein bands that you get on the gel onto the nitrocellulose membrane. So here is the difference, that in case of Western blotting, we will use the electric current to transfer the protein bands from the gel to the membrane. So the gel will be placed towards the negative electrode, whereas the nitrocellulose membrane will be placed towards the positive electrode, and then the sponge will be placed. After applying electric current, the protein bands will move from the gel towards the membrane, and then we will get an exact replica of the protein bands that are present on the gel onto the nitrocellulose membrane. After the process of blotting, we will separate the nitrocellulose membrane. Next step is blocking. Blocking is basically used to block all of the nitrocellulose filter paper except our target protein. So blocking is basically performed by using any blocking agent, or dry milk can also be used for the purpose of blocking. So why is blocking necessary? Blocking is necessary so that our probe will not bind to any other place except the target protein or any other protein, or target protein. Okay, so after blocking we will apply our primary antibody. Primary antibody is not radio labeled. Okay, so primary antibody will just bind to our target protein. Okay, after binding of the primary antibody we will wash out the nitrocellulose membrane so that the unbinded protein and other components are washed out. So after washing we will get our um, target protein that is binded to the primary. So after that we will apply the secondary antibody. Secondary antibody is basically radio labeled. So why are we using primary antibody? Primary antibody is for the purpose that we will exactly get the point where our target protein is present. So after that, secondary antibody that can bind to the primary antibody is used, and that secondary antibody is radio labeled. So after performing the secondary antibody incubation, we will detect our target protein onto the western blot or the nitrocellulose membrane that we have used in case of western blotting. So with the help of a radiograph or visualizing the nitrocellulose membrane under X-ray film, we will be able to detect the location of our target protein. Next is a comparison of all these blotting techniques. So talking about the southern blotting, northern blotting and western blotting. So first of all, target molecule. In case of southern blotting, our target molecule is DNA, whereas in case of northern blotting, RNA is our target molecule, and in case of western blotting, protein is our target macromolecule. Next is sample preparation. In case of southern blotting, DNA extraction and enzymatic digestion of the DNA is basically used to prepare our DNA sample, whereas in case of northern blotting, we use a simple RNA isolation, and in case of western blotting, simply protein extraction is used as a method to prepare our sample. Next is separation. Separation of the different fragments of our macromolecule are performed by electrophoresis in all the three techniques, whereas in southern and northern blotting we use agarose gel electrophoresis, but in case of western blotting we use SDS phage, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Next is probe. In case of southern blotting, nucleic acid probe is used that have a sequence that is complementary to our target DNA. Okay, so in case of southern blotting, we prepare complementary DNA. Next is northern blotting. In case of northern blotting, we use RNA, DNA, or oligodeoxynucleotide probe. Okay, so in most of the cases we use complementary DNA because it has a high binding affinity with our target RNA. In case of western blotting, our probe is primary antibody. Probe is basically used to detect a target protein. Okay, next, probe label. The probe is mainly radio labeled, or in case of southern blotting it can be an enzyme if if our probe is of enzymatic nature. Then we will apply a substrate, and if the enzyme will convert it into a product that might be produce luminescence. Okay, in case of northern blotting it can be radio labeled, or it can also be an enzyme, and in case of western blotting our probe label is mainly enzyme. Next is detection methods. We will use X-ray film or chemiluminescence in case of western blotting. Similarly, in case of northern blotting, we use X-ray film or chemiluminescence, whereas in case of western blotting, we use film or imaging system to detect our target protein.